All right. Well, firstly, I would like to say thank you so much to Amanda, not only for the invite, also for that wonderful introduction. When she says longtime friend, she definitely means it. She was my very first mentee, the first of about 35 now, that I have had throughout my PhD. And she kind of set the standard. So y'all are very lucky to have her. And I would just like to put that out there that I'm an Amanda supporter around here. Um, so as you said, my name is Keith. I do a lot of ecotoxicological work down Texas A&M and Gulf Coast birds. My dissertation focuses on microplastics right now, and more broadly, how we can apply bioindication methods to ecotoxicological research. Sorry. There you go. So my journey with ornithology and birds began very young. I have known I've wanted to be an ornithologist my entire life, so much so that when I was 7 years old, I forced my mom to make me a Halloween costume of an ornithologist. And I think she did a pretty good job. I mean, normally my days look a little bit different. And over the years, I've sort of curated my reputation as someone who works with cryptic species or secretive species. So I know a few of you probably know Garrett Ryan, who was a master's student a couple years ago. So I actually worked with him when I was still an undergrad, sending him some Texas samples for Swainson's warblers while I was working on a separate monitoring project. And that's sort of my first foray into hard to catch, hard to detect species. And then when I got to rails, it all went downhill from there. Um, and so, like I said, I am going to be talking about microplastics, and most of you are probably familiar with the idea of like, oh, a microplastic, it's a small plastic. Um, so just anyone want to shout out what's one thing you know about microplastics?
They're small.
They're small. All right. The other thing everyone says is they're everywhere. Um, and that's very true, right? Plastic is all over the world. It's become ubiquitous and is now both an established and emerging contaminant, which in terms of ecotoxicology makes it really unique. We don't have a lot of information on how it's affecting ecosystems and health. So it's emerging, but it is so like it's been around since the 40s and it's going to be here forever. So it is established in the environment at this point, meaning that we can tackle this like any research questions about microplastics from multiple angles. So just to start off, there is two main categorizations of microplastics, primary and secondary. So primary microplastics are the plastics that are, you know, produced at manufacturing plants, shipped all over the world to then be melted down and turned into other plastics. Could also be stuff like synthetic microfibers for clothing. And secondary microplastics are kind of what you think of as, the environmental microplastics, the stuff that breaks down from major plastic pollution. Um, and we typically group them into five primary shape classes, although it's a little bit flexible here. Fibers, beads, foams, fragments, and films. So fibers are by far the most common. They're the stuff that's shedding off of our clothes. And even though our wastewater treatment plants can filter out 90 to 95% of them, we're still getting massive amounts of fiber in our waterways, which is a massive transport system for them. And they're everywhere, like absolutely everywhere. So they've been mass produced since the 1940s with just over 8 billion metric tons in that time. Actually, between now and 2050, we're projected to produce about 5 to 6 billion more metric tons, landing us around 13 billion, which is crazy to think, considering these things are going to be around for approximately 800 to 2,000 years before they really start breaking down. So anything we produce today is going to be around much longer than we will be here as people. And they're ubiquitous. Like I said, they're everywhere. We do know that there are some habitats that are a little bit more exposed than others. So urban habitats, landfills, which are a habitat. I've gotten to arguments with people about that before. And a lot of shorelines, right? Makes sense. Anything water associated will probably be accumulating contaminants. And one of the major worrying things about microplastics and a really big point of study is that other toxicants will adsorb to them. So if you're a chemist, you'll know adsorbance properties are really big. But the best way I've learned to explain it is, has anyone here, maybe microwaved some spaghetti in a plastic Tupperware container, right? And then you have that red stain that never comes out. So that red stain is actually because the oleoresins in the tomatoes are chemically fairly similar to the lipophilic properties of plastics and the link together. And it's a little bit more difficult to remove. Other toxicants that are lipophilic will stick to plastics, bacteria, pathogens, and plastics have been a kind of theorized mode of transport for some interesting bacterial and pathogenic studies. So here's just some hopefully not too scary facts. So nurdles, right, are the primary microplastic, those beads that are shipped all over the world. Um, and to date, we've kind of assessed that about 250,000 tons of those nurdles are accidentally released into oceans every year. Until 2022, there really wasn't any monitoring for incidental microplastic contamination. So it's about all we can get is these estimates from 2016, which were externally monitored, not monitored by government or manufacturing facilities. And when I talked about adsorbents properties of plastics, if we were to test the chemicals on a plastic and the chemicals in the water immediately surrounding them, we're finding concentrations on the plastic up to 10 million times higher than the water surrounding them. So they can really hold on to those contaminants. And normally I take the piss out of Galveston County when I'm giving this presentation in Texas. I'll do South Cameron Parish. Right? Y'all have been to the coastline down there, right? You'll see quite a bit of plastic contamination. Scanning the beach, you'll see, around about 15% of the actual contamination is what you're visually seeing with macroplastics. Everything else is going to be a little bit harder to discriminate in that microplastic category. And when we think about plastics, I think it's kind of an age-old association with birds. When we think about the environmental impacts, for a very long time, we've known that seabirds will eat plastic, feed their chicks plastic, and potentially cause some disruptions in the digestive process, even starve to death or cause impaction, which is what happened and with this black-footed albatross chick here. And that's gained national attention, international attention. It's a really big seabird conservation issue. But beyond seabirds, birds in general, we have had a steady increase of publications that at least mention both birds and microplastic over the years. And when we talk about what's going on in these birds, I'm going to take you on a little bit of a journey here. I'm sorry. So we don't actually know what they're doing in the system. We have some endpoints that we can detect, right? Just some correlative, hey, we've noticed, dysregulation of estradiols. We've noticed some oxidative stress, but we don't know the mechanisms by which that's happening necessarily. And of course, there's physical effects. So after microplastics and other contaminants in the environment are ingested by a bird, they can cause some disrupted digestion, a false sense of satiation, so birds feeling full, maybe not wanting to forage so much. And in 2023, Jennifer Lauver's lab actually produced a really interesting paper calling it not fibrosis, plasticosis, just essentially fibrosis of intestinal linings that is tied to plastic abrasion. And then down the line, after they're in the GI tract and shed off what are called nanoplastics, these plastics that are less than 1 micrometer in width. Some of those will actually cross that blood barrier from the digestive tract, enter the bloodstream. And in theory, these are what's causing those hormone dysregulations, maybe some cellular oxidative stress. But we're just now kind of broaching how we can assess that. And then one other thing to note is anything that's not absorbed in the gut or, staying in the bird because it's too big to pass through the gut gets pooped out where it can then be eaten again. So again, they're lasting. And the Gulf of Mexico is actually a very interesting place to study plastics. One, because all of the plastic manufacturers, most 7 out of the 9, will actually ship out of the Houston shipping channel. So plastic manufacturers in the US use one port, And that port has a lot of incidental contamination. You can see that with these purple dots right here, centered right around Galveston County. Huge tourism and recreation area, potentially some impacts on people in those waters. And we also know that, not including microplastic, 40% of these manufacturing facilities have exceeded their limits in the past 10 years. So even if we're not monitoring plastics, a lot of these facilities don't have the best reputation for actually keeping their pollution limits below EPA established guidelines. But a lot of you probably have questions like, okay, they're everywhere. How do we detect them in samples? And if they're in birds, how do we get them out of the birds? And this is where Amanda comes in. Say hi, Amanda.
Hi.
All right. So we call this the Great Value Grey's Anatomy team, right? We're not quite as hot as the doctors on that show, but we're still wearing scrubs and white coats. I'm not as hot. So, plastic sheds off of our clothes, right? I'm wearing a plastic-based textile right now. So that means we are wearing full cotton, very scratchy scrubs, lab coats, and doing some really fun chemical analyses to figure out how to get plastic out of these birds. Amanda, what does that process smell like?
My abuela's tortillas.
Yeah, they do. So we walk into the lab and we smell fresh tortillas and cooked duck. And essentially we do this with a process called alkaline hydrolysis. And this was actually my very first year as a PhD student, was partnering with a few other research groups to come up with an assay to digest bird GI tracts down to a point where we can filter out anything but plastics and inorganic solids. I won't go into too much detail here, but essentially we use a weak base solution, a strong base, but dilute base solution, digest it, for a couple of days. Doesn't smell the best. Essentially slow cook it and filter it with vacuum filtration, where I then spend hours and hours and hours of my life looking at them under a stereoscope. So I do one pass identifying any plastics that are very obvious under incandescent lights, and then a secondary pass under black lights. And these black lights will fluoresce, or the plastics will fluoresce under black lights, depending on what their chemical structure is. So helps us to identify potentially some of the polymers, narrow it down to groups. And I'm not doing polymer analysis for my research because it's expensive, and I'm working with 10 or 15 times the amount of samples that most microplastic researchers work with. So it's just not in the 5-year plan. And like I said, microplastics are also everywhere. And this is where you can get into some trouble as a microplastic investigator. You have to correct for environmental and procedural contamination. And there's really not a good consensus on how we can do that. And so both of these limits are imperfect and probably too conservative for the estimates that would reflect a true contamination value. So I use both of them in my research. The first is just date subtraction, making blank samples for the procedure and the environment, essentially any time our sample is going to be exposed. And then subtracting that from all of the detected plastics in the samples. And then a stronger ecotoxicological kind of blanket correction is limits of detection and quantification, which is just that. Limits of detection is, when you have enough plastic to say, hey, we know that you're exposed beyond contamination levels, but we're not confident we can actually say how much you're exposed. Quantification is when we can say, hey, you were exposed to 15 micrograms of microplastics. And so I'm going to take you on a bit of a journey through my research and hopefully what I'm contributing to the field. But first, I'm going to take a big overview and talk about a framework that I'm developing to help connect ecotoxicology back into regular ecology processes. I'm going to do that through the process of bioindication. So most of you probably know what bioindicators are. Essentially, they're just organisms that we study to get measurable effects about the environment without having to study the full environment. very broad definition for a reason. These have been used for a long time. First in the early 1900s to monitor sewage effluent in rivers. And then, I mean, the process of identifying patterns in the environment and taking cues to inform our actions is a long-standing tradition in traditional ecological knowledge. And any indigenous group anywhere in the world has these values that they're detecting from the environment. So this process of bioindication has been around for as long as people have, and we still don't have a great definition for it. Even though regulatory agencies specifically recommend using bioindicator species for contaminant monitoring. And this has led to what I call the justification crutch. So I know not everybody here works with birds, but you may have seen something similar. So people who work with birds, I'm raising my hand here because this is a safe space. Who here has used the sentence, birds are bioindicators of blank, in a grant, in a manuscript? Oh yeah, did you actually talk about what they're indicating? Right? So birds are not created equally. They cover the gambit of ecological niches. So we can't say that they indicate things equally either. And a big part of this, has kind of spiraled out of control. And when you put this sentence into Google Scholar, about 35,000 results come up. And very few of them actually talk about bioindication beyond saying, hey, birds are cool, they're bioindicators. But we don't tell us why. And having a reason why and a specific framework to explain the bioindication process, what they're indicating, why they're important indicators, and also how to apply that indication process to monitoring is really needed for a lot of ecotox work. And so what I've been working on for the past few years is what we're calling a function and fit framework for bioindication, specific to ecotoxicology, but I'm collaborating with a few others to potentially expand this out to other ecological disciplines. Essentially basing the whole classification of indicators based off of their function instead of like endpoint uses. So, for example, are we investigating environmental conditions? Are we looking for early responses to change? Are we looking for changes that might be indicated in indicative of cultural shifts? So I'm going to go through these very briefly. Environmental indications, as we say, it does what it says on the tin. It tells you about the environmental condition. Right, so these are species or communities that reflect the state of the environment. And in ecotox work, most of you will be familiar with, if you are familiar with ecotox work, tree swallows. We use them because they're generalist. They tend to be robust to most contaminants. So they're not so sensitive that a single exposure is going to kill them. And we can actually monitor where the contaminants are in the environment and the state of the environment by investigating tree swallows. Conversely, early response indicators are often called sentinels or sentinel species. And if you're in disease ecology, I know it has a slightly different definition. We can talk about that later. So early response indicators are essentially the most sensitive taxa to those changes, right? They're going to be the first ones to respond and often have the most observable response. And for plastics, that's shearwaters, right? Seabirds in general, we've known for a long time, consume a lot of plastic. And we're seeing die-offs that are associated with plastic consumption. Lastly, sociocultural indicators. These are species that hold very high value in the public's view, or high social or economic value. So like waterfowl, common loons, stuff that is easily recognizable, and changes in those populations may indicate a shift in cultural values, or economic shifts in the case of waterfowl. And all of this we used to put together a whole bunch of basically a checklist of items to select bioindicators and communicate effectively about how they're indicating changes in the environment. I won't go too much into detail about that right now. I might come back to that in a bit. And so my first kind of question that I'm going to apply this to is using museum specimens as a pipeline for microplastic investigations. So I work at BRTC. We are the repository in Texas for the lights out specimens. So over the past 5 years, we have had 4,000 study skins vouchered at the BRTC, just from salvaged efforts from this big community science project. And it's way more species than any of us could have imagined. Nearly 150 species that have a bunch of different ecological value or ecological traits. And I was interested in if any of these ecological traits actually varied in the amount of exposure or helped to influence the exposure of these birds to microplastics. So it's a really big effort. We've had a ton of specimens coming through our salvage program. And then also in this data set, even though there'll probably be two separate publications, we had a really big shearwater die-off off the coast of Texas in 2023. So we had 26 shearwaters that the BRTC ended up getting that we were able to turn into specimens, including, well, not including, but also some tropic birds and frigate birds. And all of these, I was able to investigate micro and macroplastic consumption in the Gulf. And throw them into a big exposure matrix. This is a kind of a scary table, I know. Data. So running some type 2 ANOVAs and some Tukey tests for some post hoc analyses. The big thing that we're noticing is that species do seem to have differential exposure. Right? You look across this column here and you're like, oh, we have some that are exposed to absolutely no plastics or are below the level of detection, and some that are like 89% exposed. And when you look at the 89% exposed, right, when we did our Tukey tests, we averaged across all trait values and just looked between species. White-winged dove and mourning dove have very similar ecological niches, very similar ecological like life histories. Why are they so different? Why are they popping out as so different, considering all of their traits should be treated equally here? And when we throw it in a heat map, you can see that when we weight predicted exposure, so what characteristics independent of species predict exposure, habitat is the only one that is truly sticking out. So diet, we're probably seeing exposure across all dietary guilds. That makes sense. Plastics are everywhere. Foraging method, essentially whether or not a bird is using probing or non-visual methods for foraging. Again, didn't seem to play a huge role. Migratory status, we predicted that maybe stationary or resident birds would be accumulating more plastics because they are in more contaminated habitats. Also didn't show anything. So 90% of the variability predicted, variability predicted exposure can be attributed to habitat, which is really cool, right? Habitats are important. But when we compare that to our observed exposure, habitats aren't perfect. And you can see that there's definitely something more, some species-specific traits that are predicting or more important than habitat, or at least are contributing in addition to it. And this is my plea for help. Does anyone know what the hell is going on with white-winged doves? Right, mourning doves were pretty average exposure in my data set, and white-winged doves were 90% exposed. So we have some ideas about like maybe location or sort of the behavior of mourning doves maybe occurring over a broader spatial scale during different times of the year. But yeah, if anyone has questions, please, or any comments, I need help. And my second chapter is taking it a step further and going, okay, we know that we can detect these plastics in salvaged birds and dead birds, but dead birds may have died, like plastics may have contributed to that death. In ecotox, it's really important for us to look at sublethal effects. What's going on in the bird before it dies? Are these plastics contributing to behaviors or body condition statuses that are making this more likely to die? And so we need non-lethal methods. And they're not great for plastics. There's been a bit of focus on gastric lavage. also known as stomach pumping or stomach flushing, in seabirds. And it had pretty good success. On average, it recovered 75 to 95% across different populations. Um, and Mark Woodry's student, Spencer Weitzel, was able to do it in salt marsh and seaside sparrows, as well as clapper rails. But there's no data in non-seabird systems for how effective it actually is. So it's just kind of a shot in the dark. Hey, can we detect plastics with this method? So to kind of get at how effective this method is for sampling plastics, I partnered with Jen Smith at Caesar Kleberg Research Institute down in Kingsville. And she helped set up a whole bunch of ladder traps for me at Government Canyon State Natural Area, which is a big golden-cheeked warbler breeding habitat, huge stronghold. And we were removing cowbirds with the cowbird depredation program in Texas. So we were able to take these birds prior to death, flush out their stomach contents, and then test to see if we could figure out how much or what was left behind. Essentially getting at how good this is for passerines, how good gastric lavage can be. And all of these birds were vouchered at the BRTC. So big shout out to Gary Volker and Heather Prestridge for adding 200 plus cowbirds from a single date and location. And you can see that on average, it seems to be a pretty decent technique. We're not seeing differences in shapes across, either gastric lavage or the plastics left behind in the GI tract. So it doesn't seem like shape is being disproportionately sampled with this method. And it does seem like on average, there are panel A over here, lavage microplastics and total microplastics can at least be predictive of one another. Where this runs into some issues is the variability. So our coefficient of variation down here is 26. It's really high. And you'll notice that we had some that were perfectly successful and some samples that were perfectly unsuccessful. So this basically is telling us that, hey, if we're predicting individual microplastic values with this method for passerines, we're probably getting enough variation to say we don't actually know what's going on in that bird. Other than just saying, hey, it was exposed or unexposed, we shouldn't be trying to quantify based off of this. But on average, it worked really well. So this is probably a good method comparing between populations, which is a cool little use for it. We did also find that sampling date was important here, but with only 2.5 sampling dates, we sampled a whole range of environmental conditions and maybe some factors that could affect the method, whether all of sampling date was just the only one that popped out. I don't really trust that relationship with only 3 dates. So those are the 2 chapters that I have for my dissertation that are currently in prep or in review right now. Actually, I just got an email from an editor, so potentially accepted as of an hour ago. We'll see. But my next 2 chapters are a bit incomplete for a couple of reasons. So I also am really interested in microplastics in Gulf of Mexico waterfowl. I said earlier that waterfowl are good sociocultural indicators. There's a massive economic draw for the Gulf Coast to come hunt waterfowl. And because waterfowl are very habitat specific, right? Wetlands along the Gulf Coast, we could look at essentially an environmental and visual factor matrix. So this is just all of the influencing relationships that we can predict with this data set that I have up there in that diagram. And we're going to be thinking about population level ways to assess microplastics. We have to also look at differences in populations. Yeah, sorry, that was I got ahead of myself. So the state of Texas waterfowl, right? Like I said, it's a huge economic draw. There's 70,000-ish duck hunters just in Texas every year. 40% of the annual harvest from the Central Flyway comes from the state. And over 90% of the central flyway ducks will use that state at some point in their annual cycle. So the Gulf Coast of Texas is really a big deal for waterfowl. And so what we did about little baby Keith there when I was 18 years old, fit and healthy and excited about most things in life. I joined a project with my current advisor's master's student at the time, Allison, to sample a whole bunch of ducks all over Texas. And we saved all the gastrointestinal tracts from these ducks. Some of them ended up as specimens at the BRTC. Some of them ended up in my freezer because the hunters were like, here's the meat. Thanks for butchering all my birds. And so we would go to public lands and sit there in the freezing rain at the check station, hand the hunters a cup of coffee and say, you sit down, relax, we'll butcher all your birds for you if you let us keep the guts. And this allowed us to get massive sample sizes, not 2,000 from Texas alone. We have another nearly 3,000 from Louisiana. Years of my life has been spent with these birds. But this allows us to assess patterns across the entire northern region of the Gulf, which is really interesting because for microplastics, we don't have large scale data sets like that. So right now I've taken about 500 of them across a whole bunch of different species and assessed their plastics, right? So the Louisiana samples are being run by Dr. Carrie Manus at Louisiana Tech. She has been absolutely essential for all of my microplastic research and development. And so we're hopeful that with a really nice big structural equation model, we'll be able to parse out all of those environmental and trait-specific relationships with microplastic exposure. Essentially seeing if, hey, dabblers or divers have different exposure rates. Fun fact, they do. Like that's very clear. Whether or not different age classes have different exposure rates, which for some ducks, they have different age classes foraging in different areas. So we could be seeing differences there. And yeah, I mean, it's a really fun study. Unfortunately, I will also say I can't present a lot of the results of this. I'm kind of talking around it. This was originally supposed to be a really large co-exposure study with polyfluoroalkyl substances, or PFAS. And we have partners with the EPA, or formerly with the EPA, that were running these samples, and unfortunately, that has come to a very abrupt halt. And I can't present some of that data. I can say that we are seeing that there are extensive levels of contamination with PFAS in duck tissues all across the Gulf. But just some preliminary microplastics data, and this is a couple years old at this point. We are seeing differences between Texas and Louisiana populations, at least in rates of exposure. We are seeing that, about 60%, maybe a little bit less, 55%, of the ducks that we're sampling have microplastics in their gut above limits of detection. For those that are above limits of quantification, we are actually seeing a much wider range from the Louisiana populations. So while we have, in Texas, between 10 and 20 microplastic particles detected in each GI tract, we're seeing up to 80 in some of the Louisiana ducks. Now, Louisiana ducks, at least for this data, have many more species than we do. So this data, you'll learn very quickly about me. I like to do things in order. So I did all my northern pintails, all my blue-winged teals, all of that in order. So I only have 5 species from the Texas data set in that. Louisiana, we had 14 for just the scatter plot up there. So I am expecting that to expand out as I work through more species. And then, in no surprise to anyone, fibers and blue fibers specifically are by far the most common microplastic particle that we're finding. So fibers coming from clothes are the most motile, they're everywhere. What's the most common color of clothes? Blue. You look around the room, most of us are wearing blue in some capacity. So it's just interesting to note that, hey, maybe these are, some of the drivers, some of the sources behind these plastics. Lastly, and this is my heart and soul, this is what I came to grad school to do, is my clapper rail study. So Amanda knows at least at A&M, I'm known as one of the rail people. Uh, they take up so much of my life and I love it. So I talk a lot about microplastics. Microplastics are less than 5 millimeters. They're fairly easy to detect with crude methods. Nanoplastics are those really, really, really tiny ones. And nanoplastics are less than 1 micrometer in size. They are really difficult to detect. Um, and they tend to be a little bit more fragile than microplastics. So have to actually you have to have a lot more adjusting to your samples, which allows for more contamination. And we have zero contamination limits for them so far, at least in environmental and GI samples. Um, but there has been a little bit of work there. So Carl Kaiser at A&M Galveston has validated a pyrolysis GCMS method to detect plastics in tissue based off of breakdown products. And not to get too far into the chemistry, but back when I talked about those oleo resins in the tomatoes attaching to the lipophilic structures in the plastics, plastics and adipose or fatty tissues have very similar breakdown products when they're heated, which is a huge component of mass spectrometry. So being able to separate those breakdown products with lipid corrections was actually, it's really, really cool because we haven't been able to do that so far. So I'm partnering with him to look at the 12 most common microplastic polymers or NMP polymers, nano and micros, in a whole bunch of clapper rails. Just a quick note about how nanoplastics get into tissues or how toxins move through birds. We know fairly well how plastics move in the environment at this point. They've been around long enough, and there's enough study to know that at least we can trace them back to sources and we can trace those into birds. What we don't know much about is the leaching of plastics into the bloodstream and deposition of them into tissues. And as far as toxicology goes, there's a pretty streamlined process for how a bird is going to react to these contaminants. So toxins will enter the bloodstream through ingestion or the digestive process. Typically, they'll be filtered out primarily with the liver. Secondarily, they'll be filtered out through the kidneys after moving through the liver. And if they aren't filtered out by either the liver or kidneys, they'll be deposited into adipose tissue, cardiac tissue, skeletal muscle, which then could be metabolized and put back into the bloodstream. Very interestingly, and really of note for a lot of museum ecotox specimens, birds have fairly well understood molt patterns, at least up here in the Nearctic, and feathers are inert. So we can get at some more nuanced exposure of feathers once these toxins have been deposited into feathers or eggs or what have you. We can test feathers from 150, 20 years ago and look at patterns of exposure for stuff like heavy metals, potentially micro and nanoplastics if they're deposited into feathers, PFAS. So there's a lot of potential here to expand on it in museum studies. For this chapter in particular, I'm looking at liver, kidney, heart, and skeletal muscle. And I am really trying to assess the accumulative stages here. Where are these nanoplastics accumulating in the bird? Where are we seeing higher rates of deposition? And if we are able to trace those back to, very broadly, physiological patterns, we can then maybe tie in some of those endpoint physiological effects that we saw earlier, those estradiol dysregulations, oxidative stress. So we're able to figure out where they're occurring. We might be able to figure out what they're affecting within the bird. So also kind of note here, this is probably going to be the first tissue level data for nanoplastics, one in the New World for birds, but especially for the Gulf of Mexico, which there's a lot of us working on it. So I'm very excited about this. Like I said, this is my heart and soul. This is what I've been the most excited to do. Um, and so far, I've collected 26 clapper rails from 3 bay systems, but we don't talk about West Galveston Bay. Um, and I have 18 non-lethally lavaged samples to compare that with. Essentially, what are the birds like eating day of? What's in their proventriculus? What's in their crop? Um, and just comparing those values. So all of these have been vouchered as study skins. The GI tracts have been sampled and processed by me. I'm looking at different points in the GI tract for accumulation as well, particularly in the ceca, because rails have well-developed ceca. We predict that there might be some microplastics that are making their way in there and potentially staying. And I have all these tissues ready to go for analysis, and I'm hopefully in the next couple weeks going to be running down to A&M Galveston to learn how to do some mass spec and process these guys for some nanoplastics. So just to wrap up here a bit, all of my research, it might seem a bit disjointed right now, and I know I haven't talked about bioindication much in my actual projects. But overall, all of these projects are informing how we create that bioindication framework. So species and habitat level factors that we assessed in my first study with the museum specimens from the salvaged birds, we're able to say, okay, we know that we need to be looking at the species level for differences. We need to account for that in our modeling. We also know that habitat factors should be accounted for in these models as well. So if we're sampling across habitats, we should probably select bioindicators for multiple habitats. We also know that bioindication is method specific. Cowbirds as individuals, not great indicators of plastic with non-lethal methods. Whereas seabirds, 75 to 90% success rate in an individual. So we need to account for species or system level processes with the methods that might be affecting their indicator potential. And lastly, understanding movement of whatever toxicant we're studying, I'm going to limit it to nano and microplastics here, are going to help us to assess whether or not certain species or certain, ecological characteristics might, might be affecting some birds more than others. Might get us a little bit more information about how those birds are exposed, how they're reacting, and how robust they are or how resilient they are to toxicants. And if we think back to my environmental condition indicators, we wanted a really resilient species for those. We wanted sentinel species for early response, detection. We're going to want a sensitive one. So figuring out those relationships between resilience and toxicant exposure will help inform different bioindications. So just as a quick example, I'll walk you through the bioindicator selection funnel because I still got a couple minutes. So the first step of determining and using all of these findings for figuring out good indicator species is defining a research question and then selecting an indicator type based off of it. So if I was interested in, microplastic burdens across coastal bay systems, I'm interested in salt marshes or coastal bays, I'm probably interested more in environmental conditions than I am in downstream effects. So I'm going to pick an environmental condition indicator. Then, based off of all of the studies that I performed here today, I'm able to identify contaminant dynamics, which, how are they moving in the habitat, which habitats might be more or less exposed, whether or not different tissues are better or worse for sampling within these groups. We can then compare that with species natural histories, right? Species and habitat level differences. So is this going to be a good habitat, a good species for these questions? And then also whether or not there's pre-exposure conditions that may be influencing whether or not that individual or that species is a good indicator. So, we identified some contaminant dynamics. We know we want resident birds for this, so no migrants. We also want to make sure we're limiting to coastal systems because we are seeing that wetland birds may be more exposed than birds foraging in non-wetland systems. So focusing on our specialist species here, the best specialist species, I've narrowed it down to rails. So choosing an indicator from this filtered list, we should probably take a step back and go, okay, statistically, we should run a power analysis here. This is something that is not done nearly enough in ecology or ecotoxicology, in my opinion, and definitely leads to some issues when we're trying to apply toxicological data. So feasibly, what can we expect from a clapper rail versus a black rail, right? Black rail's much harder to sample, depending. A lot more legal and permitting restrictions regarding black rails. If we're testing non-lethal methods and potentially lethal sampling for some tissues, probably not going to be able to use black rails. So taking into account what we know, hey, liver tissue seems to be really good for nanoplastics. I don't know that, I'm just throwing that out there as an example. I'm not going to want to sacrifice a whole bunch of black rails, whereas clapper rails, very stable, game species, very tasty. So probably eliminating black rails, have a good estimate that, hey, clapper rails are a really good indicator for this salt marsh system. And of course we know that because clapper rails are the supreme bird species. They are the perfect organism. And clapper rail supremacy is exactly what I preach here. So I do have some acknowledgements here, but I won't go through all of them. My committee members, all of the great people who have advised me and taught me over the years, Amanda and all of the undergrads who I absolutely tortured in the field catching these birds. I'm so sorry for throwing you in quicksand and expecting you to swim. And my funding sources, so Ducks Unlimited, the Schubot Center for Avian Health, A&M, and the American Philosophical Society. And with that, I will take questions, comments, and concerns.
I'm interested in your bioindicator scheme.
Yeah.
And it sounds interesting. It sounds really useful. And as you were introducing it right at the beginning and then came back to it at the end, I was thinking that mostly down through the years when I've heard someone mention bioindicator, I thought of that as a correlate.
Yeah.
And not really anything different from that. And so I'm wondering, I get that it takes a complex process like this to figure out a good bioindicator species and to figure out a useful characteristic of that species to use as a variable to correlate with some other variable of interest.
Yeah.
So I get that. But when it comes right down to the bioindicator variable, is it anything different from a correlate?
Well, right now in its current application, not really. But it also is, yeah, I'm imagining it to be a communication framework, really, saying, hey, here's how we're going to be able to take data from potentially multiple studies, multiple systems, and finding patterns of indication. So correlates, but
I would argue that's the process to get to
yeah,
the thing that you're going to use. But the thing you're going to use, I think is a correlate.
Yeah, and that's So proxy systems are absolutely recommended, and they're not perfect, and bioindication is not perfect. But yeah, I do agree. It is heavily based on simple correlation.
Yeah, I'm not arguing against the utility.
No,
super useful, incredibly important. Right. I'm just trying
to figuring out how it's different than just saying I'm using a proxy species or a correlate. Right. And that's very valid. I've kind of through the years, that's a kind of the brainchild of multiple people on my committee who are using the term bioindicator very differently from one another, and kind of trying to figure out what they mean when they're saying, hey, this is a bioindicator for One Health systems versus a bioindicator for environmental stress versus a bioindicator for, human behavior. And so trying to just take that and at least provide some context when you're using it to kind of prompt people to give us a little bit more when they're talking about indication, I think will lead to a standard practice, or at least a more standardized practice. Yeah.
Great. Cool.
Yes.
That was great, Keith. I was wondering, so about microplastics being everywhere, across the GI tract,
Mhm.
it's a pretty uniform. For example, we have like really great stomach collection, but doesn't include ventriculus.
Mhm.
I imagine there's probably interaction there with microplastic size, but what do we know about the deposition of microplastics across the GI tract parts?
Yeah, not much. It's all been very small sample size studies that really are looking at the upper GI, the provent and the gizzard. There's not much in the lower GI or ceca or soft tissues. Definitely there are differences. So that's a big part of that non-lethal sampling study that I was doing. I know that I'm not going to get that stomach to flush all the way to the gizzard. So birds have what's called the gastric isthmus. We all do, but it's the narrowing of that GI tract between the proventriculus and the gizzard that's easy for plastics to pass into the gizzard, but very hard for them to move back out of. So in these, I'll go all the way back here to the albatross picture, where we're seeing the albatross with all the plastics that accumulated in the gizzard, that blockage that was in the gizzard, right? So the plastics can't move down into the lower GI. They're too big for that isthmus, too big to be regurgitated back out. Leads to, I skipped it. It's in there somewhere. You'll know it. Yeah, it leads to those plastics accumulating in one spot, causing that blockage. So the gizzard for sure, especially for grit-eating birds like ducks, tends to accumulate plastics and grind them down. So we're thinking that potentially grit-eating birds might have higher nanoplastic exposure as well. And then birds can regurgitate from their provent, like especially the like shearwater seabirds can take everything from their proventriculus and crop and vomit it back out if they need to. So just based off of behavioral patterns, yeah, there's probably some differences. But I really couldn't tell you other than we know they accumulate in the gizzard if there's been any patterns detected wholesale, you know. Yes.
Yes, thank you. This is a very important work that you are doing. We are completely polluted, everybody. Let's see. I was wondering whether you or anybody else had thought about testing for estrogens in vessels.
Yes.
Must be very heavy.
Mhm.
Yes, well. And you would also see whether the estrogen and estrogen disruptors and estrogen mimics would really be absorbed.
Mhm.
Because, you know, nanoplastics, yes, sure, but the real damage is done through the hormones.
Exactly.
And that would, of course, affect reproduction and growth. But it would be so much easier because you could compare with basal numbers from specimens before 1940.
Yeah. Oh, actually, I love that idea. So my advisor is an endocrinologist, and she has been really pushing me toward testing both estrogen and testosterone in a laboratory study. But I really like the idea of using pre-1940 specimens. It's a really good idea. Yeah. Well,
If you really want to go and take, you know, in January, February, March, and so on, whatever the thesis is, but it would be so much easier.
I love that idea. And I'm currently working on a grant to potentially expand on that with my advisor in the future. So,
Will you start?
yes, 100%. Yes.
Great talk. Okay, your white-winged dove, morning dove.
Yes. Oh, please, 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 please. We've been struggling with this for over a year now.
Line here. So, you know, a lot of seabirds are eating plastic because I'm sure you know this, the plastic releases dimethyl sulfur that takes their prey, so they go after it. Is it possible that plastic is releasing some other volatile that white-winged doves can smell and they're attracted to it?
That is I hadn't thought about that, but they do have a more frugivorous diet than mourning doves. Um, I wonder if volatile compounds could mimic that. That'd be really interesting. I can just add that to my list of, hey, let's start looking into it. Yes, I know you had your hand up a while. Yes.
Yeah, yeah, so what about differences if one is more vigorous? You mentioned that grid breaks down plastics or it does eat more grid. Than one.
Potentially, I don't think I've seen that. So I've taken the weight of stomach contents from both of these birds, and proportionately, it seems like at least the stomach content, the weight is about the same. But yeah, I don't know. That's another thing I'm gonna have to look into. I'm just gonna, like I said, write all these down. Yeah.
It's more about age, right? So how much do plastics accumulate over time in individual animals?
Yeah.
Is that a variable that adds to some of the variability that you're
Yeah, so that's something that we're considering with, a few of these specimens. Most of them, we'd have a good range of ages. Our white-winged doves, obviously with lights out being the source for a lot of these, they are going to be more urban. So part of our thought process is white-winged doves tend to be more stationary, roost in urban areas more often than mornings, which may be more transient between edge habitat and central urban centers. So kind of taking that a step further and going, is there age-related patterns here that might be affecting these guys? Interesting. Yes.
Pigeons, you know how the food accumulates by nano or microplastics. Because it could be that foods that are eaten by one pigeon and seeds that are eaten by the other pigeon accumulate nanoplastics differently. So in many ways, for your research, it is very good and very important. But you would really need to know how the nanoplastic gets into the food.
Exactly.
Not simply into the bird, but into the food.
Yeah, and that's a really good point, and that's something that we really don't know yet. So kind of in the same token that we don't know where they're accumulating in bird tissues, we really don't know where they're accumulating much in plants as well. We're still starting to get at that. I think the plant people are a little bit further than we are. But yeah, I think it's got to be, at least for nanoplastics, looking at whole systems and the individuals contributing to that system. So you have to look at individual plants, individual seeds produced by these plants, and then the birds eating them. So it's definitely going to be more better tested in a laboratory environment. Yeah. So it's connecting all of these back together.
Well, we're out of time.
So,
Yes.
because of the fire suppressant being off downstairs, they're gonna barricade the doors at 5 o'clock. Facility will.
The downstairs?
Downstairs, so at 5 o'clock, if you have an office down there, you need to get down there. Please, vacate before 5.
And fun fact, the fire suppressant is actually PFAS.
Well, on that note, before everyone runs to the basement, let's give one last round of applause. And if you have any more questions, we will be having the beer social on the front porch, front deck. Yeah, come join us.
Yeah.